Hi everyone, my name is Nikita Sivakumar and I'm a rising fourth year undergraduate student at the University of Virginia. Today, I'll be talking about my research using agent-based modeling and machine learning to explore multicell spheroid patterning. But first, I'd like to talk about tissue-level patterning in biology. Tissue-level patterning has been observed in a variety of developmental processes across many different organisms. An example is how in zebrafish we see these striped and spotted patterns. When we look at the neural progenitor cells that give rise to their coding, we see that they self-assemble in stripes at a very early stage in development. And a combination of experimental and computational modeling has shown that it is the homotypic and heterotypic adhesion between these neural progenitor cells that gives rise to either spotted or striped patterns. So here we can see that dynamically activated differential adhesion gives rise to unique tissue-level patterns. And we're interested in exploring how the individual cell-to-cell mechanisms of adhesion can influence the specific pattern that emerges in the tissue. To explore this, we're using synthetic cell-to-cell signaling circuits developed by Toda et al. In 2018. In these circuits, initially non-adhesive VFP-positive sender and minus-minus receiver cells engage in bidirectional cell-to-cell signaling to activate GFP-N-cadherin and M-cherry-P-cadherin cell types. N-cadherin and P-cadherin are adhesive proteins with a high level of homotypic adhesive strength. So this means that the activated green cells will preferentially bind each other, and the activated red cells will preferentially bind each other. When we co-culture these sender and receiver cells at a 1-to-1 ratio in a spheroid, we see that they form a core-pole pattern in which the green cells form a central core and the red cells self-assemble in smaller clusters around this core. The truth is, a variety of parameters in this signaling system could have given rise to this pattern. The initial seeding ratio of sender and receiver cells, as well as the sensitivity of receiver cells to sender cells, and the homotypic adhesion strength of the activated phenotypes could all play a role in what spheroid level pattern emerges in this system. Now, experimentally modifying the synthetic system over and over again to explore how all three of these parameters impact spheroid patterns would be complicated and very expensive. But we can do this with a computational model. So we developed a computational model to explore how these signaling parameters can impact pattern formation. Specifically, we developed an agent-based model of the synthetic cell-to-cell signaling system, in which we modeled the interactions of autonomous individual cell agents in a spatial grid over discrete time steps. These agents can follow rules such as moving, expressing phenotypes based on sender cells, as well as homotypic adhesion. As these agents follow rules, we can see them exhibit behaviors such as the clustering of green and red cells. And when we run these models over time, we can see the emergence of spheroid level patterns as a result of the individual cell-to-cell interactions. So here we can see that core pole pattern being recapitulated. But when we run the model for other initial conditions and signaling parameters, a wide variety of other spheroid level patterns can emerge. After developing our model, we validated it by co-culturing varying ratios of sender and receiver cells in vitro and running the 2D and 3D computational models for the same initial conditions. We've represented our 3D spheroids as X, Y, and Z cuts through the predicted spheroid. You can see that the 2D and 3D model predictions are very similar to in vitro spheroids. You can also see that as the relative ratio of minus-minus cells decreases, we go from having a central green core to a smaller green pole that's farther away from the center of the spheroid. Conversely, as the relative ratio of minus-minus cells increases, we still have a green core, but we go from having multiple red poles to just one red pole. Also, we quantified the total blue, green, and red fractional areas in vitro and in the model, and we saw that there were no significant differences between model and in vitro measurements. This shows that our ABM is accurately able to qualitatively and quantitatively recapitulate in vitro results. And so now we can use our ABM to generate predictions for new spheroids. So the first thing that we did was we wanted to explore how the parameter of homotypic adhesion strength can impact pattern formation. So this is how strongly the green cells stick together and how strongly the red cells stick together. So we varied this parameter from 25% to 100%. And you can see that when both cell types have 25% adhesion, a somewhat mixed pattern arises. When one cell type has 100% adhesion while the other cell type has 0% adhesion, these core shell patterns arise, where the more adhesive cell type is surrounded by a contiguous shell of less adhesive cells. And then finally, when the cells have maximal and high homotypic adhesion, these core pole patterns arise, which we've seen before. But these are just the extrema of this parameter space exploration. So we generated 100 samples for all 25 combinations of homotypic adhesion strengths. And we wanted to find patterns in this library of spheroids that we've generated. So manually doing that would be very exhausting and kind of biased. So what we did was we ran unsupervised k-means clustering to identify 6 different patterns. And we represented each of those 2,500 images as a dot on this dimensionally reduced space. And the color of each of these dots represents the identified pattern. And then we quantified the frequency of each of these 6 patterns in 4 combinations of homotypic adhesion strengths. So when we look at how these patterns form, we see that when the GFP NCAD phenotype has a higher homotypic adhesion strength, it's more likely for a spheroid with a green core to form. Conversely, when the M-cherry PCAD phenotype has a higher homotypic adhesion strength, it's more likely to have a spheroid with a red core. And then finally, we have these 3 mixed patterns that occur relatively frequently across these 4 combinations of homotypic adhesion. But it's more common for these spheroid patterns to occur when the first receiver cell type has a lower homotypic adhesion strength. So now we can see just by varying the homotypic adhesion strengths of the cell types, we find 6 unique spheroid patterns. The next parameter that we explored is the probability of a receiver cell type being activated by a sender cell. So this probability of expression is modeled by this function, and you can see that as the parameter C increases, the probability increases per number of neighbors with the sender phenotype. So we varied this parameter from 0.05 to 10. And once again, we generated 100 samples for each of these 9 combinations. And we ran unsupervised k-means clustering to identify these patterns. When quantifying the frequency at which these patterns occur for each activation probability combination, we see that this parameter is able to control which cell type forms the core and the number of poles in the spheroid. Using all of this information, we can use our model to design cell-to-cell signaling systems that will give us a spheroid pattern of interest. For example, let's take a case study where we want to construct a red core with 1 green pole and surrounding gray cells. So first, we need to select the higher level configuration of the spheroid, which in this case is a core pole spheroid. So we should encode both activated phenotypes in the system to have the same level of homotypic adhesion. Then, to select the number of poles, we can see that in this parameter space exploration, when the M-cherry PCAD activation probability is greater than that of the GFP NCAD phenotype, we can get red cores with 1 green pole. When we run the model with these signaling parameters, we find that the 2D and 3D model give us a spheroid with the desired constraints. So in conclusion, we created computational models of the synthetic biology toolbox and used unsupervised machine learning to explore how signaling parameters can give rise to unique spheroid patterns. And then we use this information to design signaling circuits that yield desired spheroid patterns. In the future, we'd like to investigate the mechanisms by which these heterogeneous patterns arise and validate that they occur in vitro. I'd like to thank you for watching my presentation, and I'd like to acknowledge Dr. Pierce Kotler and Dr. Lazara's labs for mentoring me in this research. I invite you to ask questions on August 6th at the live Q&A session at the Beckman Symposium. Thank you.